Okay, so what I've done is I've organized today's presentation around 4 themes. First, what I'd like to do is provide you with a little bit of the theoretical and empirical context that's been driving a lot of the questions that we've been asking, particularly over the last 5 or so years. And then what I'd like to do is just say a few words about our general methodological approach that we've taken in terms of integrating neuroimaging methods with markers of stress physiology, as well as markers of preclinical atherosclerotic disease. And then what I'll do is share some example findings from this line of research, both findings that have been published and as well as findings that are unpublished and recent. And then what I hope to do is share some of my ideas for what I hope would be encouraging next steps to continue this line of research and expand it to an understanding of how the brain might link socioeconomic inequalities with risk for other chronic illnesses. Okay, so to provide some context for the kinds of questions that we've been addressing, I'm particularly interested in the bidirectional pathways that link stress-related processes that are instantiated in the brain to risk for atherosclerotic coronary heart disease. So my overall interest is in understanding these bidirectional pathways. And to do this, in one line of our research, is this legible? In one line of research that's broadly aimed at understanding these pathways, we've been using functional magnetic resonance imaging methods to characterize some of the particular brain regions and particular brain circuits that are involved in generating responses to acute stressors. Particularly acute elevations in blood pressure and in heart rate that we all kind of feel when we're having an acute stressful experience. And a point that I'll return to a little bit later is that in the short term, these changes in physiology might serve an adaptive function. You know, they can enable some adaptive behaviors that affect aspects of cognition in the short term that are beneficial. But when these acute stress reactions are repeatedly expressed over the lifespan, they can have damaging consequences, particularly to the cardiovascular system. So for example, as I'll talk about a little bit later, people who have a tendency to show large rises in blood pressure when they're having a stressful experience are more likely to develop atherosclerotic heart disease. And what I've been interested in is the role of the brain in generating these responses. So to that end, we've been using functional imaging methods to characterize what's going on in the brain when people are showing these stress responses in an attempt to understand the role of the brain in mediating risk for heart disease. In another line of this research, we've been using structural magnetic resonance imaging methods to characterize aspects of brain morphology that I'll talk about a little bit later in the talk that are related to more chronic stressful experiences. So not these acute stressful experiences that I was just talking about, but more chronic stressful experiences. So for example, having a low socioeconomic status, and so forth, that might come to affect aspects of brain morphology over time. And part of this research is also aimed at understanding how some cardiovascular risk factors, so for example, hypertension, high levels of inflammation, and other standard risk factors for heart disease can also come to adversely affect brain morphology. And again, I'll talk a little bit about some structural imaging work in this area, particularly within the context of the study of SES a little bit later. So to take a step back and before we get into the details of research and theory kind of driving this work, I thought I would share one of my favorite stories as a health psychologist from Pittsburgh, and this could be a story that would appeal to Eagles fans in the audience as well. And this is a story from 2005 from an AFC playoff game between the Steelers and the Colts. For those of you who don't remember this game, or for those of you who are familiar with this game, it was a game that ended in really high drama. There was about, I think it was about 1 minute and 20 seconds left in the game, and the Steelers were up by 3 points. Jerome Bettis was driving for a touchdown. I think he was like at the 1-yard line. And if he would have scored, he would have sealed the win for the Steelers, and then they would have gone on to the championship game, which they eventually went on to play in the Super Bowl. So instead of scoring a touchdown, Jerome Bettis fumbled the ball, I think at the 1-yard line, and the ball was picked up by Nick Harper, who played for the Colts. And he got, I think, all the way to about midfield, and he had one person to beat. If he would have scored this touchdown, the Steelers would have lost, and they would have been out of the playoff game. And he was tackled by our quarterback, Ben Roethlisberger, who, in spite of himself, was able to make this tackle and prevent Nick Harper for scoring a touchdown, and the Steelers went on to win. And this was all very exciting, but it was way too exciting for Terry O'Neill, who was in a bar on the south side of Pittsburgh watching this game, and he had a non-fatal myocardial infarction. He literally said it was too much for him to handle, and watching Jerome Bettis fumble the ball literally broke his heart. So I'm telling this story not to make light of poor Terry O'Neill, who I think is still alive. I keep web searching to make sure I can keep telling the story. But it's not to make light of the suffering of Terry O'Neill, but it's to illustrate the difference between how I think most people think of the connection between stressful experiences and heart disease, and how researchers kind of go about studying the relationship between stress-related processes and heart disease. And to elaborate on this point, coronary heart disease, particularly atherosclerotic coronary heart disease, which is the disease process that I study, is a disease process that unfolds over many, many decades and begins in childhood. So for example, we know from studies like the Bogalusa heart study taking place in Louisiana, that children between the ages of 5 and 8 start showing fatty streaks in their arteries, which are clinically silent, but eventuate in disease processes. And these disease processes can be accelerated by a variety of factors, including health behaviors, various exposures, and then also stress-related processes that I'm particularly interested in. So my focus in my research is to kind of understand some of the factors, particularly the stress-related factors, that accelerate this disease process before the expression of clinical events like myocardial infarctions, symptoms of angina, and so forth, which are symptoms that when they're expressed, it's often very late in the disease process. Oftentimes it's too late to do anything about the disease process. And from a prevention perspective, it would be very helpful to know about some of these preclinical changes that are occurring so that you can intervene a little bit more effectively. So it's in this context that I've really been trying to understand the role of the brain in generating acute stressful experiences that might accelerate this disease process prior to the expression of late-stage events, like Terry O'Neill had. So again, to do this, we've been using functional magnetic resonance imaging to really characterize the brain circuitries that are generating acute changes in physiology, so acute changes in blood pressure that might be damaging to the cardiovascular system and accelerate this disease process. So what I'd like to do now is just describe a little bit of the particulars of this line of research. And in doing so, what I'd like to talk a little bit about is our work on cardiovascular reactivity to acute stressors as one potential stress-related pathway linking brain processes instantiated in the brain to cardiovascular disease risk. And a discussion of this will eventually turn into a discussion of how we got interested in socioeconomic inequalities and potentially the role of stress and linking inequalities to cardiovascular disease risk as mediated by the brain. Okay, so I know that a lot of you aren't familiar with this jargon term, cardiovascular reactivity. So what I'd like to do is just spend a moment or two describing what it is for you and the role of it in disease pathophysiology. Well, cardiovascular reactivity can be considered as a hallmark of a prototypical response to an acute stressor, like seeing your favorite team about to be eliminated from the playoffs, being stuck in traffic, having an argument with a friend. These are all acute, stressful events that take place over the course of a couple of minutes or so. When we're having these kinds of acute stressful experiences, most of us will show characteristic changes in our cardiovascular physiology, particularly elevations in our blood pressure, elevations in our heart rate, elevations in the force with which your heart contracts or your cardiac contractility, so that feeling of a thumping heart when you're anxious or stressed out. This is a characteristic component of a cardiovascular reaction to stress, as well as increases in the constriction of your blood vessels in your periphery, which depends on particular, which might vary depending on the particular body part that you're talking about. But these constrictions that occur in your blood vessels can help to move blood to your muscles to facilitate adaptive action. You can decrease blood flow to your gut, and all of these are in turn contributing to an overall change in your blood pressure. These cardiovascular reactions are reactions that are the end product of changes in your autonomic nervous system, as well as changes in your hypothalamic pituitary adrenal axis system, which are the two main arms of the stress response systems. These autonomic and neuroendocrine and cardiovascular changes are all the end products of changes in central nervous system activity, as shown by this naked and enraged man who's lost his mind brain over something. And it's here basically that we've been trying to kind of characterize and map out these brain systems in humans that are leading to these cardiovascular reactions that, as I'll describe in the next few slides, are reliably related to preclinical signs of disease risk. Another important point to make about this kind of stress reactivity is that not everybody shows the same kind of cardiovascular reaction to stress. So there are very wide individual differences in cardiovascular reactivity. So for example, how much one person's blood pressure goes up compared to another's, that are reliably associated with a lot of psychological and social risk factors for coronary heart disease. So for example, people with a hostile personality type are more likely to show a very exaggerated rise in their blood pressure when they're provoked by something that makes them angry compared to someone with a non-hostile or less hostile personality temperament. And then finally, probably most important from a health psychology perspective and epidemiological perspective, is that these individual differences in cardiovascular reactivity to stress are reliably and prospectively associated with a number of preclinical pathophysiological changes that are associated with later stage coronary heart disease events. So for example, people who have a tendency to show very large rises in blood pressure to an acute stressor are more likely to go on to develop hypertension, clinical hypertension in the future. They're more likely to show an enlargement of the heart called ventricular, form of hypertrophy of the heart called a ventricular enlargement. And most importantly, from my own research, I don't know how visible this is to everybody, but large magnitude cardiovascular reactions also prospectively predict the premature expression of atherosclerosis in the arteries. And in the way that most people kind of conceptually interpret these cardiovascular reactions, again, is that in the short term, they might be adaptive, they might facilitate adaptive kinds of behaviors in coping with a stressor. but when they're repeatedly expressed over the time, large surges in blood pressure can physically damage the arteries and accelerate the disease process and accumulate and the accumulation of the atherosclerosis within the arteries over time. So how do we study cardiovascular stress reactivity in the lab? Well, there are a lot of different ways that you can do this in the context of epidemiological studies. Most oftentimes what we do is we have a standardized kind of stress task that controls for individual differences in cognitive abilities. And one of the most common tasks in this area, and one task that I'll describe a little bit later, is a form of a Stroop color word interference task that has been modified to be very frustrating. For those of you who are unfamiliar with this task, the goal or the objective, if you're the participant, the goal is to identify the color in which a center word, this center target word is shown. So here, the word blue is shown in the color yellow. So what you have to do is go down here and find the word yellow, which is the correct response. And you have to suppress your tendency to want to select either blue because it matches the word, or this identifier word which is shown in the same color. And what we do in tasks like this is that we make it even harder by titrating performance so that you can never do better than about 50% accuracy. Should I answer that? Right, so basically in this way, the better you do, the harder it gets. So the trials, so basically like if you get a few correct in a row, then you have a shorter window of time to respond. So trials essentially start coming at you faster. And we do this for another reasons, because some people are better at doing this task than others, and if everyone is maintained at around 50% accuracy, then we kind of control for individual differences in performance, and then we can standardize this task in a way that can be used in multiple laboratories across the world, and then study cardiovascular reactions that might be related to disease risk. So if you have people come into the lab and rest quietly, and you measure their blood pressure, and then you have them do a task like this for a couple of minutes, and then you record the change in their blood pressure, and you study a bunch of people. So here I'm showing a little over 300 people. What you get is a distribution of cardiovascular reactivity, particularly blood pressure reactivity. So on the y-axis, we're showing the frequency of people who showed a particular change in their systolic blood pressure from rest to when they were doing this task. So on the x-axis, this is the change in systolic blood pressure from a rest period to a period of time when they were doing this Stroop color word interference task. And what you can find is that for most people, this task is not really pleasant insofar that it's causing an increase in systolic blood pressure of anywhere from about 9 to 13 millimeters of mercury. Most people are showing a rise in blood pressure. Some people, I don't know who these people are, but they're actually showing a decrease. It might be fun for them, whatever. But from a public health perspective, these are the people we're most concerned about. So the people who are showing what we would call probably a metabolically exaggerated rise in blood pressure to this cognitively frustrating task, these are the people who are at suspected risk for showing premature signs of atherosclerotic heart disease. And so what we do in our research is we link essentially the magnitude of these stress reactions to early signs of heart disease risk. The way we can actually do this non-invasively and actually pretty cost effectively now with the use of non-invasive Doppler ultrasonography. And a common measure that we use in our research, and one that I'll return to a little bit later, is a measure called intima media thickness. Intima media thickness refers to the thickness between the intimal layer of an artery, or the layer that abuts the lumen where blood flows, and then the medial layer, which is kind of this middle layer that has muscle cells in it, and it stands in between the lumen and then also this outer adventitial layer. As atherosclerosis progresses, arteries start to remodel, and the intima media thickness starts to increase. Okay? So an increase in intima media thickness is taken as a preclinical marker of atherosclerosis. And you can measure this in all kinds of vascular beds. The one that's most accessible by ultrasonography is your carotid arteries. So essentially, you put a Doppler ultrasound probe over the person's carotid arteries, and you get an image like this, and then you can go in and quantitatively and in an automated way compute the intimal medial thickness. And then you can relate measures like this to a person's blood pressure response, and you can use it as a way to non-invasively assess a person's risk and progression of atherosclerosis. A couple of other important points to keep in mind for those of you who aren't familiar with this kind of measure is that non-invasive measures like intima media thickness have been validated against histopathologic and histobiological markers of plaques and atherosclerosis and other tissue beds. And intima media thickness is a quite strong predictor of future clinical events years later, including fatal and non-fatal myocardial infarctions, strokes, and so forth. So it's a nice surrogate, non-invasive marker to assess preclinical disease, as well as a measure of being able to at least infer who's at risk for future disease events. What I'm plotting here is a very widely replicated effect and something that you typically see. So up here I'm showing kind of a sequence of events, or a sequence of the protocol where you have, you can ultrasound the carotid arteries. This is what an image of the carotid artery would look like. And then to compute intima media thickness, you're kind of computing the distance between the intimal layer and the medial layer. And this intimal medial thickness is plotted along the y-axis here. And these are data from an epidemiological study called the Kuopio Ischemic Heart Disease Study, which took place in Finland. They had a very large sample, otherwise healthy community sample, complete a battery of stress tasks, including the Stroop color word interference task that I showed earlier. And what you see is the typical positive association between the magnitude of a person's systolic blood pressure response to these standard mental challenges and the degree of intimal medial thickness. So the people who are in the top quartile of systolic blood pressure reactivity are the ones who are showing the largest intima media thickness and the ones who are showing the most preclinical atherosclerosis. And I also wanted to point out that exaggerated rises in blood pressure and large magnitude cardiovascular reactions are not only associated with preclinical markers like this, but they've recently been shown to predict premature mortality as well. So Doug Carroll and colleagues have shown that people who are showing these large magnitude rises in blood pressure to an acute stressor have an increased risk of early mortality. And again, I just wanted to return to the point earlier, is that the current thinking is that these large magnitude rises in blood pressure are probably initiating some kind of injury to blood vessels that accelerates the disease process in and of itself. So either through pressure changes or direct injury to the vascular wall, this accelerated disease process could probably come about in this way. Okay, so to transition a little bit into the neuroscience work that we've been doing, this is more of the epidemiological and health context in which we started doing our work. About a little over 10 years ago, I started to become interested in how the brain was generating these changes in peripheral physiology that have been linked to disease risk. So we began essentially by doing functional magnetic resonance imaging work to try to characterize some of the brain circuits that have been fairly well mapped out in animal models, but at the time that we started doing this imaging work, we knew actually very little about the relationship between changes in brain activity while people were having an acute stressful experience and changes in peripheral physiology that are well linked to disease risk. So in this way, we're essentially trying to characterize how we can go from an acute stressful experience to a change in brain activity to a change in peripheral physiology that might put someone at risk for heart disease. So with that, I'll say a little bit about the approach that we take in our research and then how we came to the study of socioeconomic inequalities. So again, with tasks like this, when you have standardized mental stress tasks, it has a lot of advantages insofar as you can administer a standardized task across multiple laboratories and link changes in physiology that can be evoked by this task to markers of disease risk. So when we began our imaging work, what we did is we first just started adapting these standard laboratory tasks so that they could be used in functional magnetic resonance imaging paradigms. And this is one example of a task that we started using, we developed and started using early on in our research. And for those of you who are interested, the details have been published in these papers. But essentially what we did, I'm not going to go through, I won't get bogged down in too much of the task details that are shown here. But essentially what we do is we administer tasks in what are called a blocked fMRI paradigm, in which we can simultaneously obtain blood pressure readings quite frequently while people are doing blocks of trials that evoke a lot of mental frustration and evoke a psychological state that we would probably label as stressful. And then we alternate it with, we alternate blocks of these kinds of trials with a control condition where the task is fairly easy. It's actually ridiculously easy. So where the target words are shown in a color that's consistent with all the words here, and then it's quite easy to do this task. So in this kind of intermixing, we can get a nice variation in blood pressure and a nice variation in ongoing neural activity that we can link to the peripheral physiology. And in our work, we've focused mainly on a network of brain areas that play a dual role in processing emotional and otherwise behaviorally salient information in the environment, as well as regulating peripheral physiology, particularly changes in blood pressure and cardiovascular activity, through their projections to subcortical areas that have outputs to the autonomic nervous system. So I won't go through the details of this model other than to say that we're really focusing on brain areas that play a joint role in processing emotional information in the environment and then also simultaneously regulating changes in peripheral physiology. And these are limbic or paralimbic brain areas, including areas of the cingulate cortex, areas of the insula, as well as areas, subdivisions of the amygdala. And I'll focus on the amygdala a little bit later in this talk. This is just a summary slide that was a meta-analysis or an analysis essentially aggregating all the findings of the work that we had done in this area up until 2009. And the main take-home message is that the most reliable areas that you see in terms of areas that show activity patterns that relate to the magnitude of the person's stress response are areas of amygdala as well as areas of cingulate cortex as well. So these are areas that we know are closely linked or reliably linked to a person's cardiovascular reaction that might relate to their disease risk. Okay, here's where I'll begin to transition a little bit. These are unpublished data from an ongoing study that I'll describe actually a little bit in more detail later in the talk. But what it shows on the y-axis is carotid intima media thickness. So this is this preclinical marker of atherosclerosis. And this is the mean change in systolic blood pressure from a resting period to a period of time when someone was doing this Stroop color word interference task in the scanner. Okay, so this is a stress response evoked in the scanner, and this is intima media thickness. And what you see is a little bit like the plot I showed earlier. Those people who are showing the largest blood pressure reactions are the ones who are showing the most preclinical atherosclerosis, as reflected by intima media thickness. These are some raw correlation coefficients reflecting the magnitude of the association between the blood pressure reaction and the mean intima media thickness and the maximum intima media thickness, which is another marker of preclinical atherosclerosis. And what I've shown over here are correlations between what we would think of as conventional or standard risk factors and intima media thickness, mean intima media thickness. And as you can see, one of the most robust predictors is age. And this is a healthy community sample of men and women, non-disease. I'll describe a little bit later. But the take-home message here is that the effect size of a person's stress response, a cardiovascular stress response, is about as large or on par with most of the standard risk factors other than age. So for example, being male, having a higher resting seated blood pressure, clinic blood pressure, being a smoker versus not, having elevated total cholesterol, and having an enlarged waist circumference. Also, if you can control for a lot of different risk factors, and there's still a unique association between blood pressure reactivity and IMT, insofar as blood pressure reactivity accounts for about 2 to 3% of the variance in preclinical atherosclerosis. Is this clear to everybody? All right. So now what we're essentially trying to do in our ongoing work, and what I've been trying to do for the last few years, is to really put all of this together in a mediational framework where we can essentially go from functional brain activity to a stress response to a marker of disease risk to gain a better understanding of some of these peripheral physiological stress-related pathways that might link brain with heart disease risk. So I was, very happy to do this work and to focus on this line of research, while at the same time completely ignoring, the giant elephant in the room. And I've been perfectly fine to ignore it up until this part of the talk as well. But the elephant in the room is that chronic diseases like coronary heart disease are not randomly distributed in the population. Cardiovascular disease is one particular chronic disease, like many others, that tracks the socioeconomic gradient. Okay? So to bring this message home a little bit more concretely, take the measure that's the bread and butter of my research, carotid intima media thickness, this marker of preclinical atherosclerosis. Well, we've known for over 30 years that preclinical atherosclerosis is measured by intima media thickness, tracks a socioeconomic gradient. So whether you measure SES by income or education or occupation, for the most part what you see is an inverse association. So the higher your income, the less IMT you have, the more educational attainment you have, the lower your IMT, and then also the higher your occupational ranking or higher occupational grade, the less IMT you have. There's a lot of epidemiological evidence to suggest this. And even in the scatter plot that I just showed you on blood pressure reactivity and intima media thickness, these are data from the same exact sample. And what I'm showing here is the correlations with mean intima media thickness with some standard indicators of socioeconomic status controlling for age and sex. And what you can see is the more years that you attend school, the lower your IMT. Whether you own your home or not is correlated with IMT. Your annual family income adjusted for the number of people in your household. And then also, even if you take a person's address and go to the census database and then look at the percentage of people on public assistance in your neighborhood or in your census tract, that also predicts intima media thickness as well. So at this point, when you start to appreciate data, like findings like this, you kind of have a crisis of confidence. So when I first started to appreciate findings like this, I did what most other scientists, junior scientists particularly, like myself would do, and that is, okay, well I can't really ignore SES, so I'll control for it, and I'll covary for it, in a regression model, and that's how I'll treat it. Well, when you start doing that, typically what happens is that you wipe out all the effects that you're interested in, in particular. So about 5 years ago, I decided, to not ignore the elephant in the room, and to start to explicitly focus on the role of the brain potentially in linking socioeconomic related factors to heart disease. And I think it's important to kind of put this work in context, and for those of you who aren't inequality researchers, to make just a couple of points. And those points are that chronic conditions, particularly in cardiovascular diseases, not alone, but chronic conditions like stroke, functional gastrointestinal disorders like irritable bowel syndrome, inflammatory conditions like arthritis, chronic bronchitis, asthma, as well as type 2 diabetes. All of these chronic conditions are known to track a socioeconomic gradient. Okay, so cardiovascular disease is not alone. So I think that the work I'll describe might be one potential model for understanding the pathophysiology, the role of the brain in contributing to pathophysiology underlying a lot of different chronic conditions. Another important point to make here is that risk factors for coronary heart disease and other chronic illnesses, they're also socially patterned, and this isn't going to come as any surprise to people in the room. So for example, there's very strong epidemiological evidence that high blood pressure, hypertension, dyslipidemia or elevated cholesterol, insulin resistance and glucose regulation, sedentary behaviors, likelihood of cigarette smoking, being overweight, obese, and also systemic inflammation. All of these risk factors track a socioeconomic gradient. And I think an important point to make here for the neuroscientists in the room is that all of these biological and behavioral risk factors are emergent properties of the brain and probably come to affect the brain as well in one way or another. This is a point I'll return to a little bit later. And then finally, also for other neuroscientists in the audience, there's long-standing epidemiological evidence that a lot of psychiatric disorders that are the intense focus of a lot of psychiatric neuroscience research these days, they also track a socioeconomic gradient as well. So these are data showing the likelihood of having a lifetime psychiatric disorder, comparing people with family incomes of less than $19,000 a year to more than $70,000 a year. And as you can see, for most of the affective disorders and other disorders, the odds of developing these psychiatric conditions, again, is related to indicators of SES. So again, it's in this context, about 5 or 6 years ago that I kind of started to decide to not, ignore the elephant in the room, to gain a better understanding of how facets of socioeconomic status might relate to aspects of brain function and structure that might come to influence aspects of peripheral physiology, eventuating in risk for coronary heart disease. And we started doing this research in appreciation of the fact that socioeconomic status itself is a very complex and multidimensional and multi-level construct. Typically refers, and again this is for people who don't do inequality research, but it typically refers to one standing in a social hierarchy. And it's most often thought of as something that's linked to access to a person's resources and their life's chances or opportunities in life. In doing this research, we also appreciated the multidimensional nature of SES insofar as that you have to appreciate the fact that SES can be measured by aspects of a person's education, their income, and their occupation. All of these measures can be measured subjectively or objectively, and this is a point I'll return to a little bit later. But the most important point here is that while these measures are interrelated with each other somewhat and moderately, they're not redundant and they're not interchangeable. And treating these facets of SES separately has implications for policy and intervention as well. That's the point I'll return to a little bit later. And then finally, socioeconomic status is a construct that's multi-level in the sense that you can obtain these measures at the level, of the person, at the level of the household or family, and at the level of the neighborhood, and even higher levels of analysis, county, state, and so forth. And these can all be taken across the lifespan as well. So it's here that what I'd like to do is just share some example findings from our work focusing on SES in particular and its relation to disease risk. And what I'll do is I'll begin by talking a little bit about some of one of the very first studies we did in this area that was motivated by a finding. It was specifically motivated by a finding in our lab showing that otherwise healthy adults who showed larger amygdala reactions to facial expressions that depicted threat in some way. So facial expressions that were fearful or angry. People who showed large magnitude amygdala responses to these threatening stimuli were people who were showing the most intima media thickness. So here what I'm plotting are the summary results from a study with intima media thickness that's been adjusted for a lot of other covariates, and then amygdala activation to threatening faces on the x-axis. And what you can see is those people who are showing the largest magnitude rises in amygdala activation were the ones who are showing the most intima media thickness. So what we wanted to know in one of the first studies in this area is whether we could link variation in a marker of socioeconomic status to functional activity in the amygdala, given its relationship not only to blood pressure stress reactions, but also to intima media thickness as well. And we specifically asked whether an indicator of childhood socioeconomic status is associated with amygdala reactivity to emotional facial expressions. And for those of you who are familiar, we published this work several years ago in the journal Scan. The rationale for this question came from several lines of evidence. First, there's evidence from Edith Chen and colleagues, as well as from Karen Matthews' lab, showing that lower childhood socioeconomic status appears to increase sensitivity to social threats in the environment. There's also evidence to suggest that threat sensitivity might relate to the magnitude of stress responses that might increase risk for ill health as well. And given this, we came up with the hypothesis that if lower childhood SES increases threat sensitivity, then lower childhood SES might predict increased amygdala reactivity to threatening social stimuli. And we focused on the amygdala not only for the reason I just discussed a second ago, but the amygdala is a limbic brain area that shows a lot of developmental plasticity and influence to early environmental, it's subject to influence to a lot of environmental inputs early in life. It's also probably one of the most consistently implicated areas in the processing of emotionally salient or threatening information in the environment. And then from our own work and from others' work, we know that this is an area that's involved in generating and regulating stress responses that have been linked to ill health as well. So in this paradigm, what we did is we had 33 otherwise healthy undergraduates come into the lab and complete a standard amygdala reactivity paradigm that was developed by Ahmad Hariri and colleagues. And here, what you do is you're shown an array of 3 faces, and your objective is to identify the face at the bottom that matches the center target. And the control condition in this kind of paradigm is a shape matching paradigm, where you essentially match the shape at the bottom that matches the center target. And then the way that you compute amygdala activation is you just take the difference in bold signal activity here and here. And in this study, we had the participants match explicitly threatening faces, so these angry faces, and then they also viewed and matched neutral facial expressions, and then surprise facial expressions as well. And again, the prediction here was that low SES might be related to threatening stimuli, but not other stimuli. All of the participants were first and second year college students who have not yet attained their own SES, and they filled out a packet of questionnaires that asked about the environments that they grew up in and their parents' SES. The measure that we focused on here was a measure of subjective socioeconomic status. Some of you are probably familiar with this, but essentially we had them rank their parents, their mothers and their fathers, in terms of their subjective SES. And for those of you who aren't familiar with this concept of subjective socioeconomic status, it refers to a person's perceived standing in a social hierarchy. It's most often anchored to the core dimensions of SES, including educational dimensions, occupational dimensions, and monetary or wealth indicators. And these can be referenced to a person, or they can be referenced to a person's family, particularly their parents as well. And there's a lot of evidence to implicate low subjective SES in relation to poor self-related health, a non-habituated cortisol reaction to stress. So for example, a rise in cortisol to an acute stressor that doesn't habituate after repeated exposure to that stressor. Low subjective SES has also been related to an exaggerated rise in the stress hormone cortisol early in the morning. And it's also been related to risk of having the metabolic syndrome. The metabolic syndrome simply refers to a cluster of risk factors for coronary heart disease. So there was a lot of epidemiological evidence to suggest a focus on subjective SES. Subjective SES is often measured with this MacArthur ladder of subjective social standing. And I won't go into a lot of detail here, but essentially what you do is you present people with a 10-rung ladder, and then you ask them to imagine the ladder as representing where people fall in the United States. So we say, think of this ladder as representing where people stand in the United States. At the top of the ladder are the people who have the most education, the most money, and the most respected jobs. And the people at the bottom are the people who have the least money, the least education, and the least respected job or no job. And you can say, that the people who are higher up on the ladder are better off, and so forth. And you can say, where would you put yourself on this ladder? And then in the context of our study, we said, where would you put your mother on this ladder? Where would you put your father at this ladder during the time that you were living with them before you entered college? So in this way, we got ratings of parental SES or childhood SES. And what we found, these are the results from the study, if we average across mother and father rankings, what we found is that those students who ranked their parents lowest on the subjective SES scale were the people who were showing the largest amygdala responses specifically to the threatening facial expressions. And this was not true for the surprise facial expressions, and it was not true for the neutral facial expressions. So there seemed to be kind of a selective relationship between amygdala reactivity to threat and low subjective standing. We also tried to control for a whole set of alternative explanatory factors. So when we controlled for sex, ethnicity, psychological characteristics like aspects of self-mastery, optimism, personality characteristics like neuroticism, extroversion, agreeableness. We controlled for depressive symptoms. And then what was interesting is that we even controlled for the parents' education level, which is an objective indicator of SES. And the effects still persisted for the left and the right amygdala. So this was some of the first evidence that we had in the lab linking an SES-related factor to functional variation in a brain region that we know is related to stress responses and that we know is related to preclinical heart disease risk. And so what we're now trying to do is kind of follow up on this work a little bit more formally, where we have comprehensive measures of SES. We have a lot of functional imaging measures. We also have a lot of mediators of risk, including behavioral characteristics, biological mediators, and then as well as simultaneous measures of preclinical atherosclerosis as well. So what I'd like to do now is I'll probably start winding down. I realize it's 5:30, and I just want to say a little bit about some of the findings from the other line of research where we've been using structural imaging methods to characterize aspects of brain morphology that are linked to more chronic or enduring stressful experiences, as well as exposure to cardiovascular disease risk factors. And one question that we've asked in this line of research is whether coronary heart disease risk factors link socioeconomic inequalities to the structural integrity of brain networks. And this work was recently accepted for publication in cerebral cortex, and I'm happy to share the paper with you guys by email if you're interested. And more specifically, in this work, we asked whether multi-level indicators of socioeconomic status relate to the integrity of white matter in the brain. Remember, white matter is tissue in the brain that insulates axons and neuronal tracts to facilitate conduction between brain areas. And if this is the case, if different indicators of SES are related to the integrity of white matter tracts, then what might be accounting for this association? So we focused on biological and behavioral factors that are known to relate to risk for coronary heart disease. We addressed this question in the context of an ongoing study that I've been doing since 2008. And in this study, what we did is we sent out about 36,000 flyers, recruitment flyers, to residents of all different census tracts in Allegheny County that have a very decent representation of socioeconomic distributions. So the initial phase of data collection was in 2008 to 2011, and we're now bringing all of these participants back, in addition to more people, for 3-year follow-up assessments so that we can track the progression, or at least the 3-year progression of cardiovascular disease risk. And what I'll present now are data that were collected from the first wave of this study. So the subjects that I'll be describing here were 78 men and 78 women, about 30 to 50 years old, who were extremely healthy. So they had no psychiatric disorders, they had no clinical diseases, they were unmedicated for the most part. And they completed a multi-part protocol that involved an extensive psychosocial assessment, an assessment for socioeconomic status. They completed multiple blood draws where we could assess circulating markers of inflammation, nutrient levels, lipid profiles, and so forth. They completed an ultrasound protocol for the assessment of preclinical atherosclerosis, and then they completed a structural and functional brain imaging protocol. As part of the imaging protocol, they completed a diffusion tensor imaging sequence, which is a way to look at the integrity of white matter tracts in the brain by quantifying the diffusion of water molecules. And the most common measure of white matter tract integrity, the one that I'll be focusing on here, is a measure called fractional anisotropy, or FA. So higher FA values are taken as an indirect indicator of better white matter integrity. Our multi-level indicators of socioeconomic status included education, so we use years of education. As you can see, the distribution in this sample is a little higher, has a higher skew, so most people completed about 17 years of schooling, with the range being 11 to 24 years. We also used, as an SES indicator, family annual household income that was adjusted for the number of occupants in the household. And again, we're skewed toward the higher end of the SES distribution in that the median household income was about $50,000 to $65,000 a year, but with a fairly decent range. And then finally, we took the participants' addresses, and then we went to the census database to come up with a community level or area level indicator of socioeconomic status. And this was a factor score that was a composite of the median household income in your census tract, the percentage of people on public assistance, the percentage of people below the federal poverty line, the percentage of people in your census tract who are unemployed, and the percentage of people without a high school diploma. The mediators that we looked at here are the factors that are associated with cardiovascular disease risk that in separate literatures, completely separate literatures, have been linked to indicators of SCS and also to fractional anisotropy were smoking status. We categorize people as having never smoked, formerly smoked, or currently smoke. We looked at waist circumference as a measure of central adiposity, which again is a robust predictor of cardiovascular disease risk. And then we looked at C-reactive protein as well, which is a protein, it's an acute phase reactant produced by the liver. It's a marker of systemic inflammation, and it's a very robust predictor of future clinical events. And again, all of these measures in separate literatures have been linked separately to socioeconomic status and fractional anisotropy. What we found in a very non-quantitative presentation, I have backup slides if any of you are interested in the details, but essentially we use path analyses on structural equation modeling to model the mediators going from different indicators of SCS to fractional anisotropy. And what we found is that from community SCS and income and education, the most robust pathway linking higher SCS to better white matter integrity was mediated essentially by abstaining from smoking. So those people who were of lower SCS and were more likely to smoke were the ones who were showing the lowest levels of white matter integrity. There was a little bit less robust evidence. It was still significant, but essentially in white matter tracts throughout the brain, lower SCS was associated with a larger waist circumference, and then in turn a lower fractional anisotropy indicating less white matter integrity. And then finally, for smoking and waist circumference, we found that it was C-reactive protein, so this molecular marker of systemic inflammation, that accounted for a lot of the effects, the adverse effects of smoking and waist circumference on FA. So here we're going essentially from these different indicators of SCS to these CHD risk factors, to C-reactive protein to fractional anisotropy.
Can you just point of information? Is that mediation of SES by smoking and waist circumference a full mediation? Or
partial mediation, yeah. This is partial, so this is definitely not full mediation. So overall, the main effects were that higher SES related to better white matter integrity, lower levels of these risk factors, and that these risk factors in turn, in part, accounted for some of this. And it was essentially throughout the brain. I have backup slides if you're interested in that. So this is some of the first evidence, to my knowledge, to connect socioeconomic factors to coronary heart disease risk factors to the morphology and integrity of the brain. And again, what we're trying to do now is look at this in terms of a longitudinal framework, and then also start to begin to think about questions of, when in development might you start to see some of these changes, and so forth. So what I'd like to do in these last couple of minutes is just talk a little bit about some next steps and where I hope this kind of where it goes. I think that most of you in this room appreciate the fact that SCS and inequalities in general in health, it's a very complicated field. And as I mentioned earlier, it's a construct like SCS has multiple dimensions that can be measured subjectively and objectively. They can be taken at multiple levels of analysis. All of these indicators of SCS can affect health throughout the lifespan. There are a number of different correlates of risk for SCS that span from genetic to environmental levels of analysis. But I think what's missing overall from inequality research, but also in neuroscience research, is an appreciation of the complexity and the interaction of these factors, particularly with respect to the role of the brain. So what I hope to see in this work and in future research is an encouragement of neuroscientists to have a greater interest in health inequalities. And a point I wanted to return to earlier is that, you know, most of the health outcomes that neuroscientists are ultimately interested in modeling, whether they're mental or physical health outcomes, are in all likelihood tracking the socioeconomic gradient. And I think to have a full understanding of the health outcome that you're interested in, the role of the brain in mediating that health outcome, I think in one way or another you need to appreciate you know, the SES inequalities that are determinants of the end variable that you're trying to model. What I also hope to see in this is an encouragement of more inequality researchers to have a greater interest in neuroscience. So I think in order to have a full and complete mechanistic understanding, again, of how SES gets under the skin to influence health, I think it's important to appreciate the fact that the behavior and the physiological processes that are quote unquote under the skin are emergent properties of the brain. And in some way or another can come to influence brain function and structure as well. So I think to have a complete mechanistic understanding of how SES gets under the skin to influence health, we need to consider the role of the brain. And I think in doing this kind of work and thinking about these kinds of issues, I think what we need is more interdisciplinary training. So we need more junior scientists and people who are getting involved in neuroscience and inequality research to get training so that they can speak multiple languages, so that they can speak the language of neuroscience and speak the language of epidemiology and behavioral medicine. So I do think that we need more integrative training because it's not like these problems are going away. So these are really interesting data that were published several years ago showing life expectancy rates for people who I think they were 25. So this is the life expectancy for 25-year-olds by education level from 1981 to 2000. And what you can see is that for those people who had any college education whatsoever, life expectancy increased from 1981 to the decade of 1991 to 1998. Whereas those people who had a high school education or left, their life expectancy virtually remained, it was a lot less of a steep of an increase, virtually remained unchanged. The same thing happened from 1990 to 2000, where people who had any college showed an increase in life expectancy, and for those people who had high school or less in terms of education, their life expectancy remained flat. What's contributing to these life expectancy changes? A lot of the common culprits, the usual suspects. So coronary heart disease account for about 13% of these life expectancy inequalities. Cancers, excluding lung cancer, chronic obstructive pulmonary disease, stroke, and then other factors. So there are a lot of chronic health conditions that are accounting for these health inequalities. And again, understanding kind of in the context of my own research, I've been focused on cardiovascular disease. I think there are a lot of other chronic illnesses as well as mental illnesses that we need to take into account because these health inequalities are really on the rise. So I think I'm over.